Hello friends, I am Dr. Sirajul Faizi. I welcome you in my YouTube channel. Today in this video, I am going to talk about macromolecules. So what is the difference between molecules versus macromolecules? So a molecules are monomer, a group of two or more atoms held together by covalent bonds. For example, H2O and glucose. Look at here the structure of glucose. A macromolecules or polymers, a long molecules made of monomers bonded together to form a polymer. Now you can see molecules versus macromolecules, monomer, oligomers and polymers. Now forming macromolecules form from condensation reactions called dehydration synthesis, removal of water, which is short polymers, monomers, remove water, forming condensations, longer polymers. Now forming monomers, hydrolysis reaction. Hydro means water, lysis means break. Water is added and the lysis of polymers occurs. So hydrolysis, breaking of the bond. Now memory check. Monomers, molecules, atom joined by covalent bond. Polymers, macromolecules, long unit made up of monomer unit. Polymers are formed from condensations and dehydration. Monomers are formed from hydrolysis or rehydrations. The four main types macromolecules are carbohydrates, lipids, proteins, nucleic acid. Building blocks: carbohydrate, monosaccharide, proteins, amino acid, lipids, fatty acid and glycerol, nucleic acid, nucleotides. You can see in this picture there are different types of carbohydrates: potato starch and canned sugar. So carbohydrates. Carbohydrates are the most abundant organic molecules in nature. Carbohydrates are present in human animal tissue, plants, in macroorganisms. Carbohydrates are also present in tissue fluids, blood, milk, secretions and excretions of animals. Carbohydrates are defined as the organic substance containing CHO, where hydrogen and oxygen are in the ratio 1:1 as found in H2O. The term carbohydrates means hydrates of carbon. In general formula is CnH2n ka whole n. So monosaccharide, disaccharide and polysaccharide. Classification of carbohydrate. Carbohydrate, monosaccharides, oligosaccharide and polysaccharide. Cannot be hydrolyzed into a smaller unit. Basic unit of carbohydrates. Classified based on number of carbon atoms. Classified based on the type of functional groups. Oligosaccharide, two or more monomer units. They can be further hydrolyzed to smaller unit, disaccharide, trisaccharide, tetrasaccharide. Polysaccharide on hydrolysis yield monosaccharide or oligosaccharide, homopolysaccharide and heteropolysaccharides. Now, based on number of carbon atoms, triose, tetrose, pentoses, hexoses and heptoses. Based on the functional group, aldoses, aldehyde, ketoses, keto and dihydroacetone. Now, a structure of common monosaccharide. You can see here triose sucrose, pentose sugar and hexose sugar. Triose sugars, pentose sugar, hexose sugar, glucose or galactose. You see pentose sugar ribose and triose sugars glyceraldehyde. Now a structure of common monosaccharide. You can see linear and ring forms. Now common monosaccharide: glucose, fructose, galactose. You see molasses glucose, cherry is fructose, yogurt galactose. So use of monosaccharide: glucose 50% is hypertonic and provide a source of calories in a minimum volume of water. Glucose 50% is frequently used to restore blood glucose concentrations in the treatment of hypoglycemia resulting from insulin excess or from other causes. Now fructose as an intravenous energy source for patient with hepatic disease, uncontrolled diabetes mellitus, and postoperative states. Now, dextrose is the name of simple sugar chemically identical to glucose, blood sugar, that is made from corn. Dextrose is dissolved in solution that are given intravenously, used to increase a person's blood sugar, which can be combined with other drug. Or dextrose is also available as an oral gel or tablet. Because dextrose is a simple sugar, the body can quickly use it for energy. You can see dextro energy. Now, monosaccharide of biomedical importance: sugar, clinical sugar, glucose blood sugar present in urine, glycosidia in diabetes mellitus owing to raised blood glucose hyperglycemia. Fructose corn sugar, hereditary fructose intolerance leads to fructose accumulations and hypoglycemia. Galactose milk sugar, failure to metabolize lead to galactosemia and cataract. Now friends, oligosaccharide. They are formed by condensation of 2 to 9 monosaccharide. They are disaccharide, sucrose, lactose, trisaccharide, raffinose, tetrasaccharide, stachose. The smallest and commonest oligosaccharide are disaccharides. Disaccharides consist of 2 monosaccharide units, similar or dissimilar, held together by glycosidic bond like a sugar to another groups. They are crystalline, water soluble, and sweet to taste. Now, forming disaccharide, dehydrations and condensation reactions. You can see this is the maltose are joined by a glycosidic linkage. This is condensation reactions where the water molecule is lost. Now, common disaccharide, glucose, glucose, glucose. You can see monosaccharide, monosaccharide, glucose, fructose, galactose, sugar, disaccharide, maltose, sugar, sucrose, lactose. You see the galactose. Uses of disaccharide: maltose can be converted from iodextrin, which is used in dialysis solutions. Sucrose, it is often used in medication to impart a more pleasant taste to often unpalatable chemicals. And in iron sucrose, it is medicine which is used in iron deficiency. Now, polysaccharide glycans, made up of repeating unit of monosaccharide held by glycosidic bond. During its formations, water molecule is released at each condensation. Unlike sugar, they are not sweet. So, they are ideal as storage and as a structural compounds. There are 2 types, homoglycans and heteroglycans. Homoglycans and heteroglycans. Homoglycans made up of only one type of monosaccharide monomers, starch, glycogen, dextrin, and heteroglycans made up condensation of 2 or more types of monosaccharide, agar, chitin, and chitin, and peptidoglycans. Now for polysaccharides. Polysaccharide glycans, dextrin, complex branched glucan, polysaccharide made up of many glucose. It is used medicinally as an anti-thrombic but to reduce the blood viscosity. Now, polysaccharide glycans, glycogen, storage of excess sugar in animals, stored in liver and muscles. It is the animal short-term storage form of energy. So, liver, glycogens, muscles, insulin, you can see a lot. So, chitin used as a structural materials in arthropods, exoskeleton, and fungal cell walls and surgical thread. Now, starch or amylum consists of a large number of glucose units joined by glycosidic bond. This polysaccharide is produced by most green plants as an energy store. Now, friends, cellulose, component of plant cell wall. Inulin, polymers of fructosane found in bulb, garlic, onion. Inulin is not readily metabolized in the human body and readily filtered through the kidney, is used for testing kidney functions. Now friend, general functions, carbohydrates, storage form of energy, starch and glycogen, main source of energy in the body, glycogen, excess carbohydrate is converted to fat. So structural basis of many organisms, cellulose of plants, exoskeleton of insects, cell wall of microorganisms. Component of several animals, structure and plant structure, cartilage and tendons. So carbohydrates are component of blood groups, substrates. You see, antigens. General functions of carbohydrate: ascorbic acid, a derivative of carbohydrate, is water-soluble vitamin. So derivative of carbohydrates are component of antibiotic like erythromycin, used in the treatment of infections caused by gram-positive bacteria. It is similar in its effect to penicillin. You can see the erythromycin. So friends, I hope this video will be beneficial for you. If you like this video, then subscribe my channel, and thanks for watching.